So good afternoon once again. So we are going to go ahead and look at the fundamentals of genetics. Yeah. Previously we looked at the whole process of cell division, why cells divide, the way different cells divide. We said certain cells divide by mitosis, that is to say the somatic cells, that is to say the non-gamete cells. They divide basically by mitosis, and then we said the gamete cells usually divide by meiosis. We also looked at how the genetic material that is encoded, that is within the nucleus of a cell, is lubricated during a stage of the cell cycle known as the S1 phase. So, in this study of inheritance, in the fundamentals of inheritance, we are going to be looking at how different characteristics are transmitted from generations to generations, how specific discrete packets of information are carried from parents to the offsprings. That is basically what we are going to be looking at in inheritance. So, when you look at inheritance, we cannot stop. We cannot fail to look at a man known as Gregory Mendel. Gregory Mendel is a very important man. He was a monk, a Russian monk, who went ahead and studied a garden pea plant. He lived within 1822 and 1884. He is responsible for the laws that are governing how characteristics are inherited from generations to generations. That is why we need to go ahead and look at him. So when you look at him, he was a monk and he studied inheritance of traits in the garden pea plants. That is what he studied, and he went ahead and developed the laws of inheritance that we are going to go ahead and look at. When you look at his work, he was specifically recognized. He was not recognized. In fact, he died a very miserable man, because when he first tried to publish his work, he was inspired by one of his mathematics professors at the university. They tagged him a dumb person, a very stupid person who cannot pass anything, because he tried to sit for exams to become a teacher of primary school, I would say junior secondary school, but he failed always. So when he was so disappointed, he went ahead and started doing some small experiments about the garden pea plants, and he was studying how characteristics are inherited in the garden pea plants. When he wrote his book, during a time like that, you could not write any book. You could not publish anything unless it has gone through the hands of professors. So he had to go to one of the professors, and they refused to publish his work on grounds that they all believed that he was dumb and there's nothing good that could come out of him. His work was acknowledged to be to carry a lot of information after people had studied about inheritance using the fruit fly, and that is in the 20th century. That is when he was recognized for his work. But he died a very miserable man, despite the fact that yes, he came up with a lot of information that we are using up to now. This is this gentleman called Gregory Mendel. So he resorted to being a monk, and that is how he was given a small piece of land in the monastery, and he tried to do his experiments about the garden pea plant, and even he documented his work. And that tells us as scientists that yes, as you are coming up with new ideas, you always need to document your work so that it can be of use to those who will come there after you. So that is Gregory Mendel. When you look at Gregory Mendel, so he did his experiments between 1856 to 1863, where he cultivated about 28,000 pea plants. He found that the plant offsprings retained traits of their parents. This was his first observation that he made. He observed that in all the different generations he cultivated of the garden pea plants, he realized that the offsprings tend to retain characteristics of the parents, and he is so considered to be the father of modern genetics because of his work that he developed. So putting that aside, so sites, where did he carry out his experiments? Czech Republic. You can see that area, that is in the Czech Republic. That is where he used to carry out his experiments. He was given a small garden. So putting that aside, that simple history, we can go ahead and look at the particulate inheritance. Mendel stated that physical characteristics are inherited as particles. That is his observation that he made. He said that characteristics, or when you look at the different physical characteristics that we are seeing right now, he said that they are inherited as particles. Mendel did not know anything about about chromosomes or DNA. For him, he just said we usually inherit certain particles from our parents, and they are the ones which are accounting for the characteristics that we exhibit, the reason as to why we resemble our parents. He didn't know anything about the DNA or the chromosome, completely nothing about them. So we can start by looking at some of the terminologies that were used or are used in genetics. To begin with is what we call a trait. Any characteristic that can be passed on from their parents to the offsprings, that is what we call a trait. Take an example: tallness or dwarfness. Any inheritable characteristics or any detectable variant that you can carry on from parents to offsprings, that is basically what we can call a trait. It can be tallness, it can be dwarfness. Heredity is just a study or a science that deals with how characteristics are carried from the parents to the offsprings. Then genetics is simply the study of genetics, the study of inheritance, the study of how characteristics are passed on from generation to generation. That is what we usually call inheritance. So we are going to add on more terms here. So we can add on more terminologies here. Another term that we need to know is a character. What is a character? We have looked at a trait. What a trait is. We said a trait is simply that specific feature. We said a trait is that inheritable characteristics that you can pass on from parents, offsprings. That is what we call the trait. Now, within a trait, we have we also have what so a character. We have another term that we can use: a character, inheritable feature. So when you look at a character, like for instance, height of the stem, that is a character. This height of the stem can exist in two different traits. That is to say, it can be tall or short. That is the trait within this character, height. The character is a height. And then within the character we have traits, character traits. The traits are shortness or tallness. Okay, so the next thing that we can look at, we have looked at a trait, a true breed. We need to look at a true breed. A true breed are all offsprings of the same variety, offsprings of the same variety. And at this level, we shall use terms like homozygous, offsprings that are showing which are homozygous for a specific, the offsprings which are homozygous for a specific character, or what we usually say capital T, or small t, they are homozygous. Both chromosomes are carrying the same kind of gene. Then we shall say that is a true breed. And a true breed, when you allow it to inbreed with each other, they produce the same kind of breed without any changes. That is what we call a true breed. There is what we call hybridization. In hybridization, we usually say when you get two pure breeding parents, let's say parents who are purely tall pea plants. When you get purely tall pea plants and you cross them with purely short pea plants, what you form in between is called a hybrid, which is heterozygous, as we are going to refer to them. We are going to see the meanings of each of those terms. So when you get two organisms which are having the same, when they are homozygous or they are pure breeds, two pure breeding parents, and you interbreed with you allow them to interbreed, that process is called hybridization. So we shall look at the different thing. We usually take an example. What do we mean by hybridization? Take an example of this individual here, capital P crossed with small p, and then you get capital P small p. This one here is a hybrid, what we usually refer to as a heterozygote. These pure breeding parents, for them they are referred to as the homozygous. They are homozygous. Homo means the same type of allele. There are other terms that we have not seen here. Terms like allele. A gene usually exists in two forms. or in 3 forms depending, but normally most of the genes exist in 2 forms. The gene can exist as a capital letter or a small letter, like this. Considering this gene here that we are having here, this gene that is responsible maybe for color of the petals, you can see that it can either be capital P or a small p. So these different forms of the same gene, they are the ones we usually call allelomorphs, or allele or allelomorph. Allele is these are the alternative forms of a gene. So if both homozygous chromosomes are carrying the same type of allele, then we shall say that is homozygous. If the homologous chromosomes, both of them are carrying 2 different types of alleles, let us say one of the homologous chromosomes has capital T, capital P, another one has small p, then we shall say that one is heterozygous. So an allele is simply an alternative form of a what? Of a gene. Okay, so putting that aside, now we are going to go ahead and see, so we can go ahead and look at other terms like a generation, P generation. When you see the word P generation, what should always come to your mind is it is the parental generation. When you see the word F1 generation, the F1 generation, like for instance here we are having the F1 generation is this heterozygote here. So the F1 generation is the generation that is produced as a result of a cross between a hybridization cross between 2 pure breeding parents. What you get is what we usually call a cross or a hybrid. That is the F1 generation. Then F2 generation, or what they call the second filial generation, it's a generation that is obtained when we get these F1 generations and we do what is called selfing. We allow them to breed with each other. It's like allowing the sister and brother to produce children. That is what we call selfing, and that is what we call the F2 generation. As we are going to look at his experiment, we shall see, and we shall see and appreciate very well. So you can see here, so the F1, the P generation is the parents here, pure breeding purple, pure breeding white. The F1 generation is the heterozygote. The heterozygote is the F1 generation, which is a hybrid. The F2 generation are those ones which are produced when we get the F1 generation and we allow them to interbreed with each other. What we get is called the hybrid. What we get is the F2 generation. So let us go ahead and look at the different types of genetic crosses that we can make. We have very many different types of genetic crosses that we can make. So we have what they call a monohybrid cross. In a monohybrid cross or monohybrid inheritance, the word mono means one. So when we are crossing, like for instance, pure breeding parents, let us say flower color that we have seen in our previous example. When you're looking at flower color and you are only looking at flower color, that is to say you're getting pure breeding parents who are red, who are purple, and pure breeding parents who are white, and then you cross them. This type of cross is known as monohybrid cross. You are not looking at inheritance of other characters or factors. You are only looking at inheritance of a specific single trait, how it is inherited. Then that is monohybrid inheritance or monohybrid cross. You are crossing a pair of contrasting characters, but of the same, a pair of contrasting characters, but for a single trait. That is to say, flower color. You are looking at how purple and white are inherited. You are not focusing on the length of the internodes. You are not focusing on the texture of the pods. Now, having looked at that, we can go ahead and look at, before we can go ahead and look at this square, there is also what we call dihybrid cross. In a dihybrid cross, you look at 2 traits at a time. Let us say you look at flower color. How is flower color inherited along with another trait? Take an example of maybe stalk, length of the stalk. That is in dihybrid inheritance. You look at 2 characters at 2 traits at a time. That is say flower color, and then 2 maybe then you also look at inheritance of the length of the internodes. So having looked at that, a Punnett square, we need to understand there are a number of tools that we shall use when we are doing genetic crosses. we also need to understand how the Punnett square works. So when we are making genetic crosses, we usually use a tool. The tool that we usually use is known as the Punnett square. This Punnett square usually helps us to solve the genetic problems. How would we draw the Punnett square? We usually make a small square that is of the same length, same width. After they are divided into 4 different sections. Okay, so after dividing into the different sections, the 4 different sections, the next task is you are going to fill in the gametes. The gametes are usually placed on the top and on the side of the squares. Take an example of what we are having here. So you put the female gametes on top here. That is to say, one correlating to the first square, then the second one correlating to the second square, but on top. Then you put the male gametes on the side, one correlating with one square here, then this one correlating with this square here. There you will have drawn a Punnett square. Then what comes next thereafter is you are going to do what is called fertilization. What I usually tell my learners to do is you drop the letters. You drop the ones which are on top, you drop them through, you put this P that is here, you put it here, then you also put it here. Then do the same for this one. You put this one here and put it here also. Then after doing that, you drag these ones which are on the side from left to right. That is to say, you are going to put the P here and the P here also. Do the same for this also. P here and P here. That is how you end up with this small P here and along with another small P here. That is what we call fertilization. So you are getting the F1 or the F2 genotypes. So how to make the Punnett square? I've told you it allows the geneticists to predict possible genotypes and phenotypes of the offsprings. Let us take an example where we are having heterozygotes who are parents who are heterozygous for yellow P allele. The first thing you do is to place the alleles of the gametes on the parents along the top of the grid and along on the other side, on the left side of the grid. The second task you do is to combine the parent alleles inside the boxes. The letters show the genotypes of the offsprings. You can see we have done it now. After doing so, you will have what we call the genotype. Now you go ahead and find out the genotypic ratios. Okay? How? Now we are going to go ahead and look at this law of dominance. I don't want to look at it now, but we shall look at it, the law of dominance. So when you do like that, you fill them in. Now, you use the law of dominance. Now you will go ahead and say, now, how many capital Y do I have? I have 1. So that is why I'm having a ratio here, 1. Then how many capital Y, small y do I have? I have this and this. That means I have 2. Then how many small y do I have? I have only 1. That is why we have a 1 to 2 to 1 genotypic ratio. Now this ratio here is a genotypic ratio. And now when we have this genotypic ratio, we fill in the offsprings. We usually use the dominance to determine the phenotypes and the phenotypes and the phenotypic ratios of the offsprings. So in this example, we can say that the one that is dominant is the one for yellow. So that is why they are saying even when you are capital Y, small y, all of them will appear to be phenotypically yellow. Why have we chosen this to be yellow, to be the one to take the dominant or the one to take the capital letter? Why? Because when you look at the parents, all the parents were appearing yellow, yet they were heterozygous. When you have heterozygous and all of them are showing a certain characteristic, as we are going to see in the dominance cross, you will always take the one that is expressing itself in the F1 generation to be the one that takes the capital letter. Always remember that the characteristic or the trait that is expressing itself in the heterozygous state, only the F1 state, F1 generation, we call that the dominant one. Okay, so let's go ahead and look at the gene. We have said the gene can exist in 2 different forms. It can exist in a dominant form or recessive form. I've already talked about that. Those alternative forms of a gene are called the alleles. It can exist as capital letter or a small letter, capital P, small p. So the dominant gene or the dominant allele is stronger of the two genes and is usually expressed in the hybrid, and it is represented with a capital letter. I've already told you about that one there. I've told you that the one that is usually given the capital letter is the one that expresses itself in the hybrid. Take an example in our previous example. The one that is the hybrid parents that we are having were all showing yellow. So that means the gene for yellowness is dominant over the gene for green. So it takes the capital letter. It is the one that even expresses itself in the first filio generation. I told you the first filio generation are the hybrids produced from a cross between two pure breeding parents. Two pure breeding parents, I would say, pure breeding green and pure breeding yellow. When you get those two and you cross them, you will get a hybrid. So if you get two pure breeding parents, pure breeding yellow and pure breeding green, and you cross them and you end up with yellow. Yellow is one of the parents. So the one carrying yellow, the character or the letter that is going to carry yellow will take the capital letter because it has expressed itself in the hybrid. Then the recessive gene is usually that shows up less often in the cross, and it is usually represented with a small letter. In fact, for it, it will show up again in the second filio generation. Okay, that is when it will show up. It cannot express itself in the hybrid. In fact, when the capital Y letter is around, the small letter is silent. It will not do anything. It does not express itself. It only expresses itself when both of the homologous chromosomes are having the small letter. That is when it will express itself. But if it is not like that, for it will never express itself. When the capital letter is present, whether it is on only one chromosome or it is on two chromosomes, that one will only show the dominant allele. The one, if we are having the small letter and small letter, then we shall have the recessive gene being expressed. Now, putting that aside, we are going to go ahead and look at some more terminologies. A genotype is a gene combination for a given trait. A gene combination for a given trait is what you call the genotype, or the genotype is the genetic composition of an organism. The kinds of genes that an organism is carrying, that is what we call a genotype. The kind of genes an organism is carrying is a genotype. An organism can have capital R, an organism can have capital R, small r, an organism can have small r. And then we have another one that we call the phenotype. The phenotype is simply the physical appearance of an organism, how the organism looks like. And the phenotype is produced as a result of an interaction between the genes that the organism is carrying along with the environment in which the organism is surviving. So a combination of genotype and the environment, they bring about what we physically see. When the genotype, the genes are saying this one is capital R, then the environment is also in respect to that, we end up with what we call a phenotype. Capital R, then the phenotypes we shall have words like red, white, that is the phenotype, the physical appearance of an organism, and it is the manifestation of the genotype of the organism and its environment. That is what we call phenotype. So we can go ahead and look at the different phenotypes and genotypes in flowers. Take an example, genotypes of alleles. When we have capital R, that is red flowers, small r, yellow flowers. We are just assuming, but in a given example, in an exam, you might find that the yellow one is the one that is taking the capital letter. But just reading the question very well and understand it, and then you attempt appropriately. I told you the one that takes the capital letter, the word that takes the capital letter is the one where it is expressing itself in the first filio generation. For instance, if they are to give you an example and they tell you, we had a cross between yellow flowers, pure breeding yellow with pure breeding red, and all the F1 generations were all red, then that tells you that the red is the one that is going to take the capital letter. Okay, so you can see the possible combinations, the possible genotypes we can have of these two. We can have capital R, small r, capital R, small r, small r. So the phenotypes, you can see in the phenotypes now we shall have capital R, and capital R small r being red. Why? Because the capital letter does not allow expression of the small letter when it is around. So the dominant characteristics are the ones which can express themselves in both the homozygous state, this is homozygous state, and the heterozygous state. But when you look at these other ones known as the recessive characteristics or recessive gene, for it can only show up its characteristic when it is in homozygous state. The dominant one can express itself in both homozygous and in heterozygous condition. That is what we call dominance. Okay, so you can see now we go ahead and look at now the genotypes. Homozygous, I told you homozygous genes composition involving two dominant or two recessive genes like capital R, small r, capital R, or small r, and they are also called the pure breeding. The heterozygous, I told you, a genotype or a gene combination where we have both of the different kinds of the gene together. That is to say, both of the alleles are present. The dominant allele is present and the recessive allele is present. That one is called a hybrid. We talked about that. So genes and environment determine the characteristics of an organism. So when the genes interact with the environment, they give us what we call a phenotype. Now let us go ahead and look at Mendel's experiments with the Pisum sativum, or the garden pea plant. Pisum sativum was chosen by Mendel because of a number of reasons. But why? That's the question. Why did Mendel, among all plants, why did he choose Pisum sativum? That is a very wonderful question that we have ahead of us. We need to try to answer. One, Pisum sativum can grow in a very small area. You need a very small area to grow Pisum sativum. And when you look at Pisum sativum, you will realize that it also produces very many offsprings. It can grow in a very small area, it produces very many offsprings, and it produces pure parents when allowed to self-pollinate for several generations. It produces pure parents when allowed to self-pollinate over several generations. And it can also be artificially cross-pollinated. You can artificially cross-pollinate this Pisum sativum. You can do it by manually, like the way Mendel was doing it. Other reasons are this Pisum sativum tends to exhibit very many different traits or very many different characteristics that we can study. Take an example. When you look at it, you will see that we can study about the length of the internode, we can look at the color of the petals, we can look at the color of the pods, we can look at the texture of the pods, we can look at a diversity of different characteristics about this Pisum sativum. So being able to have very many different characteristics that we can study about makes it a very good example, and it's having a very short lifespan. Since Pisum sativum has a very short lifespan, that means that we can study very many generations within a short period of time. Imagine we were using a human being who has a life expectancy of about 65 years, or maybe considering Uganda, 65 years, a lot of years. That means that for you to study the offsprings of this human being, you will have to take 65 years. That is a lot of time. A scientist cannot live for more than the average amount of the life expectancy. So for Pisum sativum, it had a short lifespan. It can produce very many offsprings. It can be grown in a very small area. It has very many characteristics that we can study about, and it produces pure plants when allowed to self-pollinate for very many generations. Now, when you look at reproduction in plants, if I'm to give you a preamble about reproduction in plants, I will tell you that pollen usually contain the so-called, in quotes, sperms. They are usually produced by the stamen, and the ovaries contain the eggs, which are inside the flower. And now those ovaries are the eggs, the ovules. So usually pollen contains the sperms and the eggs. The pollen contains the sperms, and the egg that is within the ovary, they are usually fertilized during the process called double fertilization. So this fertilization, we can have self-fertilization or we can have cross-fertilization. In self-fertilization, or what they commonly call self-pollination, the pollen grains of the same flower pollinate the same flower. Pollen grains land on the stigma of the same flower and cause it to pollinate, to fertilize. That is self-pollination or self-fertilization. Then another type we usually call cross-fertilization, which occurs between different flowers. Okay. So what this man known as Mendel used to do, he would simply, Mendel's method usually was hand pollinating. He would get the pollen grains from a desired plant, and then he goes and he would cover up the stigma of the flowers, of his flowers that he's using to study. After covering up the stigmas of the flowers he's using to study, he would go ahead and hand pick the pollen grains from the stamen of a desired plant, and then he comes and pollinates the flowers. That's how he used to do it. To prevent self-pollination, that's how he used to do it. He would carry them with a paintbrush and transfer them. So he traced traits through several generations. Now, how Mendel began. One, he had to establish what we call pure breeding parents. That is it, the first job that he had. He had to establish what we call pure breeding parents. How was he establishing pure breeding parents? For instance, you've gone to the market and you have bought Pisum sativum seeds. You are not sure that they are pure breeding parents, or they are not pure breeding seeds, because among them you might find those ones which are yellow, others are green. So what he did, he allowed them. What he did, he planted them, and he made sure that he prevented cross-pollination from taking place. He ensured that they self-pollinated each other for very many generations. Now, when he ensured that they did self-pollination of each other for very many generations, that is when he was able. So Mendel produced pure strains by allowing the plant to do self-pollination for several generations. That is how he was able to attain what we call the pure breeding parents, by allowing them to do self-pollination for very many generations. There are very many traits that Mendel studied, but he studied about majorly 8 of them. That is to say, the seed shape, the shape of the seeds. He looked at round versus wrinkled. He looked at seed color, yellow versus green. He looked at the pod shape, smooth versus wrinkled. He looked at the pod color, green versus yellow. He looked at the seed coat color, gray versus white. He looked at the position of the flowers, that is to say, axial versus terminal. He looked at the height of the plant height, or the length of the internodes, where he looked at tall versus short. He also looked at the color of the flowers, purple versus white. Those are the 8 traits that he was able to study: seed shape, seed color, pod shape, pod color, seed coat color, flower position, plant height, and flower color. Those are the different traits that he was able to study. Now, so you can see some of them here. You can see all of them here. He looked at the smooth seed. You can see how it looks like: wrinkled, yellow, green. Pod color, pod shape, you can see inflated, wrinkled. You can see the color of the pods, green and yellow. He also went ahead and studied the flower color. He saw purple along with the white. The stem, flower position, axial versus terminal, or at the tip. He went ahead and looked at the length of the stem, short, tall, and then short. So experimental results. What did he observe? After doing some research, when he crossed pure breeding parents, that is to say, the ones which were pure breeding for smooth seeds, take an example. I'm just going to take an example of smooth seeds and wrinkled seeds. Okay, I can take yellow and green seeds. He observed a ratio of 3 to 1 ratio, or what we usually call a 3 to 1 ratio. You can see among all these ones that you are having here, it is approximately a 3 to 1 ratio. You can all attest with me that all of his offsprings in the F2 generation, he observed that all of them on average had a 3 to 1 ratio. You can see from the one for texture of the seed coat, the yellow, the color of the seed coat, the texture of the seed coat. You can see all of them had a 3:1 ratio. That was his experimental results. In the F2 generation, he observed that all of them were showing a 3:1 ratio. That is a point to think about. Why were they showing a 3:1 ratio, whereby one character was carrying three, on average was having 3, and then the other character was only having 1. Those were his results. Did the observed ratio match with the theoretical ratios? The theoretical ratios he expected were, for instance, the round and wrinkled seeds expected 3 round and 1 wrinkled. So when you look at, for instance, a ratio which is 2.96 to 1, the discrepancy is due to statistical errors. Sometimes maybe you have problems with the reading, but when you have a very large sample size, then it is approximately the same as the theoretical ratio. So the difference is very small. Then we can go ahead and look at the generation gaps. We looked at the first filial generation. We looked at the parental generation, which is labeled P. Then we looked at the F1 generation, which is known as the first filial generation, produced as a result of a cross between 2 pure breeding parents. You get the hybrid known as the F1 generation. We looked at the F2 generation, and we said it is the second filial generation, where you have selfing of the F1 offsprings. The hybrids, you get the 2 hybrids that were produced from the F1 generation, and you self them. When you self them, what you produce is what we usually call the F2 generation. Okay, so we can see the following generations. You can notice with me that in the P generation we had pure breeding tall and pure breeding short. When they were crossed, all the F1 generation were all tall. And you can all attest with me that when we crossed the tall ones together, we ended up with 3 tall and 1 short. This is the 3:1 phenotypic ratio that I was talking about. So this is 3:1 phenotypic ratio. This is of the F2 generation. This is Mendelian inheritance. You get a tall pea plant crossed with a short pea plant, you end up with all tall in the first filial generation. And then when you self the first filial generation, you end up with 3 tall and 1 short. Okay, so you can see that clearly. So a cross of pure breeding parents will give you capital T, capital T, crossed with small t gives you capital T, small t, a hybrid, but it is showing a dominant character. And then the capital T, small t crossed, you will end up with capital T, you will end up with tall and then short as phenotypes, but in a 3:1 ratio. Monohybrid crosses, we can look at the monohybrid crosses. I told you about the Punnett square. So take an example of a trait that we are looking at. We can look at seed shape, round or wrinkled. So when you're looking at seed shape, round or wrinkled, when we are the parental phenotype, this one is our parental phenotype, round seed crossed with the wrinkled seed. Our parental genotype will be capital R, small r. This parental genotype, when we do now fertilization, during fertilization we put the male gametes on the right, on the left, and then the female gametes on the top. After putting them on the top, now we are going to use a Punnett square to carry out our fertilization. So during fertilization, what we are going to do is, that is our first genotype offspring, the genotype of the offspring. Then the second one, the third one, and the fourth. So those are our offsprings, the F1 genotypes. That is our F1 genotype. So our F1 genotype is capital R, small r. Our F1 phenotype is round seed. All of them are round. And now our genotype, our genotypic ratio is all alike, and the phenotypic ratio is all alike. That is a representation. For instance, they are to tell you to explain the observation, explain Mendelian observation in the F1 generation. What do you do? This is what you do. You tell us, let capital R represent round seeds, let small r represent wrinkled seeds. How do you tell which one is going to take capital and which one is going to take small letter? I told you, read through the question. When you read through the question, they will give you a clue. They will tell you that when round seeds were crossed with wrinkled seeds, all the F1 generation were round seeds. That is enough clue to tell you that it is the one that takes the capital letter. Now, now we are going to go ahead and look at now monohybrid cross review. Okay, we have done that. So homozygous dominant crossed with homozygous recessive, that is the phenotype. All the offsprings are heterozygous, or they are hybrids. Offsprings are called the F1 generation. The genotype and the genotypic and the phenotypic ratio is all alike. Now let us go ahead and look at the monohybrid cross, where we are going to go ahead and look at. So that's what we have been looking at. We have seen a cross, how we make a cross. When you're writing it, you start with the parental phenotype, that is to say round seeds crossed with the. So now we are going to see our selfing. For instance, when we are selfing our F1 generation, what do we obtain? When selfing the F1 generation, what we are going to obtain is, we are going to obtain, you can see now that now we still repeat that let capital R represent round seed coat, or round pod, or round seed. Then let small r represent wrinkled. Now the parental phenotype that we are having now here is the one that was produced from the F1 generation, which is round seeds crossed with round seeds. Capital R small r crossed with capital R small r. That is what we are having as now our parental genotype. Our parental genotype is this, parental genotype is this. Parental phenotype, new one, is this, as a result of what we call selfing. So, after doing that, now, so we fill them in. We fill the male gametes here and the female gametes here. Okay, there we are. We have filled the gametes very well. Now we are going to do fertilization now. This will go with this, so we have capital R there. Then this will go with this, then we shall have capital R small r. Then this will go with this, then we shall have capital R small r. Then this will go with this here, then we shall have small r small r. That is our now we have produced our F2 genotypes. Those are our F2 genotypes. So the genotype of the F2, we have capital R, small r, capital R, and small r, small r. So the phenotype now is going to be round seeds and wrinkled seeds. And the genotypic ratio is different from the phenotypic ratio. You can see that the genotypic ratio is 1 to 2 to 1. But remember, because we are bringing in the principle of dominance, we are having capital R, it is 1. Capital R, then capital R small r, there are 2. Then small r, there, it is only 1. Okay, so we have a 3 to 1 phenotypic ratio, but a 1 to 2 to 1 genotypic ratio. Now, we are going to, so reviewing, we had 2 heterozygotes. Heterozygotes produced from the F1 generation. And the offsprings were 25% homozygous for the dominant character, that is to say capital R, capital R. 50% were heterozygous for the that is to say capital R, small r. 25% were homozygous, that is to say small r, small r. Now, they were the offsprings. So the offsprings that we had, those ones are the ones which we call the F2 generation. Now, when you look at the phenotypic ratio, we had the genotypic ratio is 1 to 2 to 1. The phenotypic ratio is, now our phenotypic ratio is going to be 3 to 1. Why? Because the capital R small r and then capital R, both of them are existing as they are showing the dominant character, and that is round seeds. So now what do the peas look like? That is the question that we are going to go ahead and answer. That is how they look like. When you open the pod of the peas, that is how they look like. You can see some of the peas have a smooth texture, while others have a wrinkled texture. You can see this one is smooth, this is wrinkled. This is smooth, this is wrinkled. You can see that this is smooth, wrinkled, smooth, wrinkled. You can see that some of them are smooth, others are wrinkled. And now we have a question. We have said that the capital R, small r exposes the dominant character. Then capital R, small r also shows dominant character. capital R, small r also shows the dominant character. So we ask ourselves a question: how then can we know that this one is a pure breed and the other one is a heterozygous? That one, Mendel was able to solve that problem by carrying out what he used to call a test cross. A test cross is carried out to ascertain the actual genotype of a suspect who is exhibiting a dominant character. A culprit could be exhibiting a dominant character. So when a culprit is exhibiting a dominant character, we don't know whether he is homozygous for the dominant character or heterozygous for the dominant character. So what we usually do, we do a test cross to ascertain whether he's dominant for that character or whether he's heterozygous or homozygous. How? We get that one that is being whose genotype is unknown, and then we cross him with a pure breeding parent. But this pure breeding parent who is showing that showing the recessive character. Take an example. If we have tall and short, the one that is showing the dominant, the shortness is recessive. So we get homozygous for short, or homozygous, let us say wrinkled. And then we cross him with the suspect. With so doing, we shall be trying to ascertain the actual genotype of this suspect. That is what we call a test cross. We want to ascertain the actual genotype of the suspect who is exhibiting dominant characteristics. He might be homozygous or heterozygous. So you get that aspect and you cross him with a pure breeding parent, and preferably the parent that is exhibiting the recessive character. Now, so we can have our okay. So here, preferably here, what was used here, what we have here is we want to find out whether the suspect is homozygous or recessive, or heterozygous. You get the one who is pure breeding. So you can see in this example we have pure breeding round, and then the suspect who was produced from the F1, who has the F1 generation. Who is this? So when you get them and you cross them, the offsprings that you will get, you can fill in you can see that now we are filling we are filling in the gametes. Please draw that square and then try to fill in. Pause the video and then try to fill in. Pause and try to fill in the gametes, and then try to come up with the genotypes, the expected genotypes. Pause the video, come up with the gametes and the expected genotypes, and then go ahead and also find out the genotypic ratio and then the phenotypic ratio. Pause, do that. Okay, I believe you have all filled that, and I believe now you are now confirming your answers. So these are the gametes, we have filled them. Now, after filling in the gametes, now we are filling in the genotypes, capital R, then we have this with this, then we have small r, capital R, okay, and then we have this capital R here. This will go with this, so we shall have capital R, then we shall have this with this, capital R, small r. Okay, so there we are. Now, we have the genotypes. We have, now, how do we know that this one who was a suspect was homozygous? We shall know that he was, okay, we shall know that he was heterozygous for the dominant character. Take an example here. When all the offsprings, in fact, when all the offsprings are showing the dominant character all around, then we shall say that this suspect here that we had was homozygous was homozygous for this specific example. But if we had used small r here, we would get a mixture of both round seeds and wrinkled seeds, and that would tell us that the suspect is heterozygous. I repeat, if we had used the recessive parent here as our test cross and the suspect, and you go ahead and make a cross and you find that all the kids, among them you have round seeds and wrinkled seeds, that is enough to tell you that the suspect was heterozygous. Now, how do you tell that the suspect was homozygous? You can tell that the suspect was homozygous if you had a small r here and then small capital R. All of the offsprings will be round. Then you will say that the suspect was homozygous. That is what we call a test cross. Okay. So this is what I've been trying to explain. This test cross here, that's what I've been trying to explain. If you have small r as you are the one who is known already, cross him with this one here, you will see that you can fill in the gametes. You can see we can fill in the gametes, fill in the gametes. You can see we have filled in the gametes. So the gametes are here. So here we have capital R, small r. Capital R, we can have the genotypes now. We have capital R, small r. Then we have small r, small r. Then we have capital R, small r. Then we have small r, small r. When we have that, then when you have in your genotype, you have capital R, small r, and then small r, then you have the phenotype will be round and wrinkled in a ratio of 1 to 1. Phenotypic ratio 1 to 1, genotypic ratio 1 to 1. That means that now the suspect was a heterozygote. Okay. So, we can do some review. So homozygous crossed with the heterozygous or the hybrid. If you have 50%, 50%, then the phenotypic ratio 1 to 1, then we shall say. So you can try out this. Try out this simple exercise. You can try out this one here. Work out the F1, F2 of both the crosses for any of the. Choose one of your choice. Choose one of your choice and then work out. I think it can take you about 2 minutes. Choose any of your choice. You can choose length of the stalk, tall and short. Work out the F1 and the F2 and then tell me. Okay, we can go ahead and look at the laws of inheritance or Mendelian inheritance. That's what we are going to go ahead and look at. This gentleman came up with about three laws, but we shall only look at 2. When you look at his results, we saw inheritable factors or genes. For him, by then, he was just calling them factors. He didn't know that they are genes. So, it is Wiseacres who called them genes after some time. So, inheritable factors or genes are responsible for the inheritable characteristics. That is, those are his results. Then he said phenotype is based on genotype. Then each trait is based on 2 genes or factors, one from the mother and one from the father. Those are all his observations. Then true breeding individuals are homozygous. That is to say, both alleles are present at the same time. That's what we call homozygous. When you have capital R, or small r, small r. So the law of dominance, that is the first law of inheritance. That is what they call the law of dominance. That is the first law that he came up with. In a cross of parents that are pure breeding of a contrasting characters, only one form of the trait will appear in the next generation. All the offsprings will be heterozygous and they will express only the dominant trait. In other words, let me give you just a simple term. I will tell you that all characters if I'm to rephrase the law, it says that all characters in living organisms are controlled by a pair of factors, but only one factor exists in each of the gametes. I repeat, all characteristics in living things are controlled by a pair of factors, but only one factor exists in each of the gametes. Why is it so that only one factor exists in each of the gametes? We talked about meiosis and we said during meiosis the homologous chromosomes separate, and one of the homologous chromosomes goes to one cell and the other one goes to another cell. That is how we can explain how it comes about. So the law of dominance, you can see it here. So 3 genotypes yield. You can see all these 3 genotypes, all of them, but they are only yielding yellow and green. Only 2 phenotypes. That tells us that we have 2 different genes. A characteristic is controlled by 2 genes or 2 alleles. That one for yellow and that one for green. But remember that of those 2 genes, only one exists in each of the gametes. Okay. Then the other law that is known as the law of segregation. They usually say that during the formation of gametes, two alleles are responsible for the characteristic. Okay. The first law we said, the law of dominance, that always we have two alleles. Okay. But one allele known as the dominant allele will always cover up the other one that is known as the recessive allele. So the dominant allele will take the capital letter, and then the recessive allele takes the small letter. Then the law of segregation, sorry about that, I think I mixed them up earlier. When you look at the law of segregation for them, they say that during formation of gametes, all characteristics are controlled by a pair of factors. But only one factor exists in each of the gametes. Now, alleles form a trait and then they are recombined at fertilization, producing genotypes for the trait of the offsprings. I told you about that one already. So they say a character is controlled by a pair of factors, but only one factor exists in each of the gametes. But then they recombine after fertilization. Those are the processes that we have been doing. So today we have been looking at Mendelian inheritance, so you can see how the law of segregation is applied. You can see here we have a female parent who has capital T small t, but because the gametes only carry one, she will release them into small t capital T separate gametes, and then the male parent also the same. So you can see here, and then after fertilization, now we form a genotype having both of the alleles. That is how the law of segregation is applied. So that is it. Next time when we meet, we shall look at the law of independent assortment, and we shall also go ahead and look at the different characteristics that are inherited in a Mendelian fashion in human beings. So we shall look at some examples of Mendelian inheritance, and then we shall look at some deviations from Mendelian inheritance. So today we have only looked at Mendelian experiments and Mendelian laws and how they are applied. The different new terms that we look at in inheritance, we have also seen them. So that is basically the introduction of inheritance in living organisms as per Mendelian inheritance.